Mutations in the DNA. On the basis of the severity of the damage, silent mutations are very less, causes the damage which is very less than the missense mutations, and which is less than the nonsense mutations, and which is less than the damages by the frameshift mutations. The types of the point mutations are transition and transvariance, whereas point mutations are the changes in the single nucleotide. Transition is the type of mutation in which there is purine to purine or pyrimidine to pyrimidine exchange, and transvariant is the type of mutation in which the purine is replaced by a pyrimidine or a pyrimidine is replaced by the purine. The single nucleotide mutations, these are silent mutations, missense mutations, and nonsense mutations. The silent mutations are the mutations in which the nucleotide substitution codes for some amino acid which is same as the previous one. Often the base change in the third position of the codon, and when the base change in the third position and there is no change in the no change in the amino acid, then it is called the RNA wobble. Missense mutations are the mutations in which the nucleotide substitution results in the changed amino acid called the conservative if the amino acid has the similar chemical structure. Examples include the sickle cell disease, in which the substitution of glutamic acid takes place with the valine. And the nonsense mutations are those mutations which result in the early stop codon UGA, UAA, UAG, usually results in the non-functional protein. We can say it by mnemonic stop the nonsense. Other type of mutations are frameshift mutations and splice site mutations. In the frameshift mutations, there is deletion or insertion of a number of nucleotides not divisible by 3, like you can see here. Misreading of all the nucleotides downstream. Due to this insertion, it leads to the misreading of all the downstream nucleotides. Proteins may be shorter or longer, and its function may be disrupted or altered. Examples include Duchenne's muscular dystrophy and Tay-Sachs disease. Splice site mutations are the mutations in which the introns are retained in the mRNA protein with the altered function. During the splicing of the DNA, after the DNA formation, the introns are not removed in such type of mutations, and these examples include rare cases of the cancers, dementia, epilepsy, some types of beta thalassemia, Quecher's disease, and Marfan's syndrome. Thank you.